This lecture is a brief overview of bioinformatics. Some of the software and databases we'll be using in the lab portion of the class. What you see here is a workflow for transposon localization. So imagine you have a population of bacteria, it could be Vibrios or Salmonellas or whatever, and your goal is to create a library of random transposon mutants, and therefore you can dig into that library and study the phenotypic effects of each individual mutation on the particular mutant strain. So you could, for example, screen the entire library for defective biofilm formation, and then any strains that have defective biofilm formation relative to the wild type can be sequenced to localize with a transposon hit. And then once you localize with a transposon hit, you can start to do work on that particular gene because you've already got a phenotype for it, which is the aberrant biofilm formation that I mentioned. And so again, this workflow for transposon localization begins with genomic DNA extraction from transposon mutants. And again, usually you're picking the mutants that have interesting phenotypes. So another example might be you could take your entire library of mutants and screen each one individually for defective motility. You know, something easy that you can quickly see on a simple assay is ideal. So in this particular case, imagine we've isolated a mutant that is defective in cell division. And so that means that the cells, rather than forming individual rods or vibrios, they form chains, which is very unusual for that particular species. And so right off the bat, you can say this has an aberrant cellular morphology and arrangement. So then we would do the genomic DNA extraction from the transposon mutant. So here's our pile of genomic DNA, and here's a theoretical location of gene X. And remember that the two colons indicate that a transposon was inserted in this particular case. So here's our transposon, here's gene X disrupted by that. We can then cut we would then cut the DNA with this enzyme SAU3A1, and that would create these pieces as you see here, cutting the transposon, essentially bisecting it. And the reason why SAU3A1 is used is because we know where the SAU3A1 restriction cut site is, and we know that it's a pretty frequent cutter in the genome. And so we'll get cuts all over the place in the genome, but we know that it will only cut once in the transposon, essentially bisecting it. If we self-ligate those pieces, as you see here, so we basically just add DNA ligase, and then these pieces will, because the ends are sticky, they've been cut with the same enzyme, so they leave sticky ends that can self-ligate. We can circularize these as indicated here. So each of these fragments, including the fragments without the transposon, all the fragments that get cut in the genome can be self-ligated into circles like this. And then we can do something called inverse PCR or iPCR. And so in that case, we use PCR primers that look away from each other, and we get amplification, linear fragment amplification with the transposon on one end of the amplicon, and then the gene of interest here. And so then we can use a pyrosequencing technology. So again, we have a primer that sits right here and reads outwards in the sequencing reaction. So it'll basically just give us this bit of sequence right here to tell us where our transposon inserted. And so once we do the iPCR, the pyrosequencing with these primers that sit here on the transposon and look outward and essentially polymerize outward so that we can get this bit of DNA sequence, we can then compare that to a database like BLAST and identify the particular insertion location. So in this hypothetical example, the insertion location was in an amidase gene. And so then we can ask the question, does it make sense that an amidase mutant should form chains as opposed to single rods or vibrios? And the answer is yes. There's lots of precedence for that. Amidases tend to cut peptidoglycan during the cell division in order to separate cells. And so again, it's a neat little closing the loop type experiment. So this is just a little bit about the technology for pyrosequencing. We use this technology if you take if you ever end up taking Bio 475, the molecular biology lab, or Bio 501, if you come back as a grad student, or if you're already a grad student, you'll perform this technology, pyrosequencing, to localize transposon insertions. And so the technology is actually really neat. The chemistry is very neat. And so imagine here's the direction of synthesis. Here's a piece of DNA that we want to sequence. And so each time a deoxy GTP, for example, or any deoxynucleotide gets incorporated into the chain, it's dephosphorylated. And so as the polymerase is going through and reading this, the next gene or the next base in the sequence would be a G that it needs to pair with this C. So in this example, the deoxy GTP is incorporated and the hydrogen bonds are formed here. It's dephosphorylated, so here's the pyrophosphate group, reacts with APS in the chemical reaction in the machine. APS, in the presence of this enzyme, ATP sulfurase, will make ATP. So out of this combination, APS plus the pyrophosphate, in the presence of ATP sulfurase, will make ATP. And the chemical reaction also contains a little luciferin, which is, essentially the firefly luciferin that's necessary for light production in fireflies. There's also a little luciferase in there, and so this combination of ATP, luciferin, and luciferase yields a little bit of light. So imagine a nucleotide. So there's other nucleotides in the pool as well, like DATP, for example. So what the machine will do is first feed a DATP over here, and that won't get incorporated, and so it gets dissolved and broken down. The machine will feed a DTTP, and that will get dissolved and broken down because it's not incorporated. And then eventually it'll get to DGTP or whatever nucleotide base is required, and that will be incorporated. So whatever gets incorporated releases the phosphates and ultimately releases light. And so the computer, the machine rather, the pyrosequencing machine, is actually looking for these pings of light. And each time it gets a ping of light, after trying out the 4 different bases, whichever base emits the ping of light, that base gets recorded here as the growing sequence. And so this over here is basically just showing you that that reaction making ATP, and then the ATP combining with luciferin to in the presence of luciferase to make light. And so that's what's pictured here again. And then this reaction here is showing you how those unused nucleotides, the DTTP and the ATP and so forth, don't get incorporated, and they end up getting broken down, so they don't hang around by this apyrase enzyme. So by this basic technology, the machine will give you a DNA sequence for the flanking region to the transposon. And so again, this region here. And you can use some software like Finch TV, for example, to view these chromatograms. And so each of these peaks, then, the highest peak represents the base that was incorporated at that particular position. And you can go through and see how clean the sequence is in this particular case. But sometimes sequences are awful messy, like this one here. This is a low quality sequence that I would not trust. Again, you can see some peaks that are sort of overlapping, and the machine just makes a judgment call, essentially. So if we see something like this, we know that something went wrong with the sequencing and not to trust this DNA sequence. But something clean like this is great. And again, if we submit this to a database for comparison and identification, odds are we'll get a clean match. If we submit this, we'll get a bunch of low quality matches that aren't statistically significant. So BLAST is one of the database, basic local alignment search tool. It's one of the databases that we use. Here in the class, we'll use another database called KEGG, which has BLAST incorporated into it, the algorithm. And so what does BLAST do? Well, it's a family of programs that allows you to input a query sequence. And so again, the query sequence can be DNA or amino acids, and compare it to DNA or protein sequences in an extensive database. And so here's the URL to get there. And usually the homepage looks sort of like this, and then you can choose your particular database or your particular algorithm that you want to use. Mostly you'll use nucleotide BLAST, but you can use a couple others like BLAST-X, for example, which is a translated nucleotide that will you'll give you data, do a search for proteins in that database. In T-BLAST-N, you can submit a protein and get a translated nucleotide, and then a straight-up protein BLAST where you just submit an amino acid sequence and get a matching one back. And usually it's an unknown amino acid sequence or it's a known amino acid sequence, and what you get back is all the homologs of that. And so if it's unknown, you can use this to identify it. And if it's a known sequence, you can use this to figure out who else has a gene similar to that or a gene product similar to that in other organisms. And so here's a typical interface for the BLAST-N. And so here's the information box. And so you would put your DNA sequence here, for example, and then you would choose the database. Typically, we use this nucleotide collection database. And if we're working with bacteria, we generally don't want to deal with mouse or human genomic DNA. And then we can optimize for highly similar sequences. If we have a really good sequence that's really long, we can optimize it, and so we can only get back really significant hits. And then again, there's other parameters down here that we can modify, but that's generally these basic parameters work very well, and will get you the information you need. And you click BLAST and receive your homologs. If you need help, there's a BLAST glossary that you can go through, but just some of the glossary terms. An alignment obviously is the process of lining up two or more sequences to achieve maximum levels of identity. Identity meaning matching, and conservation. And so again, this is alignments are the key here. That's what the database is doing. It's taking your query sequence and aligning it to millions of sequences in the database, or hundreds of thousands, or whatever, however developed that particular database is. A domain is basically a discrete portion of a protein that's assumed to fold independently of the rest of the protein and possesses particular functions. So we can actually take an amino acid sequence and do domain searches to see if other well-studied proteins exist, and to give us an idea of what domains they share, and that might help us understand the role of an unstudied gene, for example. It's pretty amazing how much work we can get done without even lifting a micropipette these days, given these databases. You can do a lot of dry lab work and learn a lot about a protein without doing any biochemistry or molecular biology these days. One of the things that we're looking for is the E-value, or expectation value. It's essentially the number of different alignments with scores equivalent to or better than S, and that's a particular score, that are expected to occur in a database by chance. And so the point is that when we get an E-value for a particular alignment, the lower the E-value, that is, the closer it is to zero, the more significant the score is. When an E-value is zero, that means there's a 0% chance that your sequence aligned to this other one just by random chance. And obviously, the greater that number is, the more likely your sample is to have been aligned by chance. A gap, as the name suggests, is if the software has to create gaps in the alignment because it's not a perfect match, and therefore that affects the score. That affects the score quite a bit in terms of, and ultimately affects how the E-value is determined if there's a bunch of gaps. If the sequence aligns well, but there's gaps that have to be added, then that obviously it still suggests possible homology, but also some divergence or possibly even sequencing errors. So it looks something like this. Here's a gap. Again, there's alignments here, and then there's obviously some mismatching. This particular sequence doesn't line up. So A, G, A, C, there's a mismatch, and then T, G, A, and then it jumps over to C, T, G, A. So there's a gap that had to be added, suggesting again that this organism's sequence is different from this organism's. Or it might also suggest that there was a sequencing error. And so what you can do, one thing you can do is go back to your chromatogram and find that location and then see what happened. Is there a potential? Is the sequence garbled in that area? Now, if it looks like this, it's nice and clean, then no. But if it's like this, then there's a decent chance that there was a sequencing error. HSP, high scoring segment pair, local alignments with no gaps that achieve one of the top alignment scores in a given search. And so again, we're looking at high scoring segment pairs in this particular case, and I'll show you the total output in a minute. Identity is the extent to which two nucleotide or amino acid sequences are invariant, meaning that identity, this is these guys, 1, 2, 3, 4, and these 4 bases, they are 100% identical. The identity is 100%. A low complexity region can affect the score as well. You may have a high level of identity, you're meaning that meaning that there's a lot of identity here. There's a lot of alignment. But if the sequence is low complexity, like for example, long stretches of A's or long stretches of T's or something like that, the software is sophisticated enough to recognize that and say, okay, well, it's a low complexity region. We're not going to give that a very high score. So the query sequence is your input sequence, and that's the sequence that's going to be compared to all other entries in the database. And what's amazing is the time with which that happens. I mean, the software is so sophisticated that it can do these queries in seconds. The raw score, as I already mentioned, S, is the score of an alignment. It's calculated as the sum of substitutions and gaps. So any mismatches or gaps that have to be added affect the final score. Similarity is a little different than identity. It's the extent to which nucleotide or protein sequences are related. And so, again, it reflects the fact that, for example, when you're comparing amino acid sequences, amino acids fall into groups, polar amino acids versus non-polar amino acids. So if the amino acids are not identical, the software will also look at, okay, if they're not identical, if they're not both prolines, for example, are they then both hydrophobic amino acids or hydrophilic amino acids or acidic or basic or whatever? If they're similar, then you still get a positive score for that, but not as much, obviously, as if it were the exact match. So, and then at the nucleotide level, similarity is considered based on, for example, the wobble effect, where the third position codon, again, we know that there's multiple codons that can encode the same amino acid. And so just because two organisms have a different codon doesn't mean that those two different codons aren't still encoding the same amino acid. And so the software can actually recognize that wobble effect. And therefore, even though it's not identical, there's still a similarity score that gets factored in. And again, substitutions, as I've been kind of alluded to over here, is just when the presence of a non-identical amino acid at a given position in an alignment. If the aligned residues have similar physicochemical properties, the substitution is said to be conservative. So as I mentioned, so sometimes it's outright, totally different amino acid, polar for non-polar, whatever substitution. But in many cases, the substitution is subtle, polar for polar. And therefore, it affects the scoring in a different way, as I said, than if it were completely different. And so, again, there's lots of specialized searches that you can do. Smart Blast, for example, finding proteins that are highly similar to your query. there's a bunch of different things. A primer blast, you can submit a DNA sequence and have the software tell you what are some good examples of primers if you're trying to decide on how to do a PCR for that particular hunk of DNA that you want to amplify. So lots of cool examples of that. Some of the additional features, you can actually align two or more sequences. And so if you click that, then instead of just submitting a hunk of DNA and having it compared to a database, if you click align, you'll get a second information box, and then you can align to any two sequences in your own possession to figure out if they are homologous or similar in any way. And so again, here's the appearance of the BLAST N window. So here's your DNA sequence. You can select nucleotide collection database and highly similar, click BLAST, and you'll get sort of an output like this, and some of it's color-coded and graphical. So here's your query sequence. It's goes from 1 to about 456, as you can see here. And then the query sequences, the sequences that were returned by the search, the top match aligns from here to here, suggesting that it's not a perfect match because there's bits of it that are missing, but because again, your query sequence is 456, and here's where the top match in this particular case aligned. And so again, this is the top match, and then here's some actual data. This query coverage is 66%, so 66% of this guy matched this guy here. That's your sequence. And the e-value is 2 times 10 to the negative 73. So because this is even though this is a short sequence alignment, 66% coverage, it matched up so well that the e-value is infinitesimally close to 0. Not quite 0, but very close to 0. So we know that it's significant. We know that this is real, that whatever you submitted is really close to this section of the Vibrio fischeri chromosome, for example. And then you can click on the succession number. The accession number will take you to the particular alignment and as well as the annotation for what that particular gene probably is in the chromosome of Vibrio fischeri. In this particular case, N-acetylmuramoyl L-alanine amidase. So again, that's the AMI-B or amidase gene. And you can get a sense for how closely related. You can see some gaps here and mismatches and so forth. But you get this general sense that overall the alignment there's 84% identity. There are some gaps, but the bottom line is the expectation value is so low that it suggests that your sequence your unknown sequence is definitely this gene. And it also suggests that you may have some sequencing errors that cause these problems, unless this is from a different organism, in which case these could be real, and that your sequence just diverged from the Vibrio fischeri sequence. And so you can definitely link to the different references for this thing, and again when you click on the accession number, it'll take you to. So in this particular case, and I think I misspoke here, but you can click on the score here and go to this, and then then you can click on this, the accession number, to get to this information about that particular gene sequence in the database. And so here's the source of the organism, here's the paper that it came from, the journal that it was published in, and so forth. And so you can get a lot of information from that. And then if you scroll down further from here, you can get this type of information as well. What the gene name is, AMI-B, the locus tag, how to find it, and how it's annotated in the Vibrio fischeri genome. And then here's the full amino acid sequence from the initial methionine here to the end. And then you can do additional resources, you can use additional resources to do author searches and in PubMed or whatever to find all the papers you want about this particular gene. And another database that you can use is KEGG as well, and that's one that I primarily use more so than BLAST. KEGG is a little bit more specialized and has a little bit better, in my opinion, user interface. But you can still do a BLAST, so you can still start with a BLAST. So if I click this BLAST right here, not the FASTA, but BLAST, it'll take me to a BLAST search window, and then I can input my sequence here in this dialog box. I can tell it whether it's a protein, like BLAST P, or if I want to do a BLAST X, which is a nucleotide query that will be assayed against a protein database. And so it'll basically just translate your nucleotide to protein and then return protein homologs. But in this particular case, if I wanted to do a nucleotide blast, I'd click blast N or whatever. And the bottom line is I input my sequence, choose the appropriate database, and then I can select a few more specific parameters here. I can actually tell it, if this is a blast N, for example, I can tell it what organism I want to search if I want to search a particular organism. Or we can be a bit more specific and tell it that I want to, if I pick this category, I can pick a prokaryote, for example. And so again, the idea here is that you can be highly specific rather than having the software chug through all kinds of non-specific stuff. You can be more specific, and you can put in a favorite organism code or a category. So again, if I know it's Vibrio fischeri, you just have to kind of know these codes, but you can click on that and figure out what it is. And category can be prokaryote or eukaryote or whatever, but the organism code, since this is Vibrio fischeri, I would type in VFI, which is the code for Vibrio fischeri. If I wanted Vibrio cholerae, VCO, or E. coli, ECO, or so on and so forth. But anyway, the point is you can be highly specific and then compute, and then I would get a return of sequence alignments. And here's the top scoring one, again, the MEB, and then the E value, 6 times 10 to the negative 25, which again suggests that it's a highly similar match. And then I can get some other examples of homologs and other microbes. And again, you have to kind of know what these different abbreviations are. But here's VFI, Vibrio fischeri, and then the particular gene annotated in the genome. One of the cool things about this, though, is that I can then click on this in KEGG and get to a really great informational page on that particular gene, VF2326 in this case. The gene name is AMIB, and the definition is N-acetyl-muramoyl L-alanine amidase. So this is an amidase gene essential for cell division, basically cutting up peptidoglycan during cell division, allowing the cells to actually septate and physically segregate from one another. The organism that it came from, there's a whole bunch of other things, and you can look at hierarchies for the gene and different motifs within the gene, within the amino acid sequence. And so again, you can see that there's an amidase motif, lysam, and amine motifs. And so going down, you can see that there's a genome map that you can click on, and that'll show you where the gene is in relation to other genes. The question is it in an operon? Is it all by itself? What direction is it pointing in the genome? You can get a lot of really cool information from the genome map, again, without even lifting a micropipetter. The full amino acid sequence and its length, and then the full nucleotide sequence and its length. And so you can even ask for upstream and downstream information. So if I wanted to know, so here's the start, the start codon is theoretically over here, and the stop codon is right here. But what if I wanted to know what the promoter looked like? I might, and I don't know exactly where the promoter is, but I know that it's upstream of this. So I would ask for 200 nucleotides upstream and get all those nucleotides, and then I can study that DNA and clone it potentially and and study where the essential RNA polymerase and other transcription factor binding sites are. And so ultimately, when you have a transposon mutant, getting back to what I showed you way up here, like this, we can, and then ultimately getting to this point, using these tools, I can figure out exactly where the transposon inserted into the gene. So imagine this MEB gene is 1728 bases long. Oh, here it says 1731, but anyway, I might have changed it a little bit. So it's about 1730 bases. Pretty big gene. But the question then is, where did the transposon insert itself? Remember, these transposons can be 1,000 to 2,000 base pairs. In our case, the transposon that we're working with, TN170, is about 2 kilobases. And so that's pictured here. So these 2,000 bases stuck themselves in between base 182 and 183. Now, this is obviously, it's not drawn to scale because this is technically longer than this, so, but I just put it in there like that for simplicity. The bottom line is this transposon insertion hit fairly early in the gene, and the reason why we might want to know where the transposon inserted is, again, to figure out whether or not we can expect a full mutation or a partial expression of the gene. Imagine if this transposon inserted way back here and disrupted the tail end of the gene. We might still get 80% of the protein made, and therefore some version or some form of function, that some amount of functionality. And therefore the gene might still work a little bit, and therefore our mutant might not be as ideal. But in this particular case, the mutation smacked early in the gene, and so therefore we can expect that the replication machinery wouldn't be able to make anything but a bunch of junk. And so again, this kind of is a brief overview of some of the bioinformatics software, and in our particular case, how we'll use them in the lab.